It's my pleasure to introduce Nicole. So Nicole Tignor has been really the driving force behind this next project called Project Hope, which is a kind of collaboration between CPTAC and CPTN. Nicole is, as I mentioned, the driver, but a key member of Pei Wang's group up at Mount Sinai, which is one of the CPTAC centers in data analysis. And we've just been so privileged to work with Nicole and her group to help wrangle this multi-omic data into some semblance of order, which she's going to show you.
Good afternoon. In this talk, I'll share findings from a collaborative CPTN and CPTAC proteogenomic study of high-grade gliomas in adolescents and young adults. Gliomas in adolescents and young adults, as defined by the age range of 15 to 40, are typically diffuse, with anaplastic astrocytoma and glioblastoma being the most common. Despite extensive research, these high-grade tumors remain largely incurable, and patients in this age group are relatively understudied. In our study, we analyzed 93 high-grade glioma cases in this age group using a proteogenomics approach. We found that DNA changes and molecular profiles were different depending on age and sex. We also analyzed kinase activity, which helped us to identify some potential drug targets. Finally, we identified distinct immune signatures linked to protein clusters with poor outcomes. We've characterized the tumors using 6 omics types: whole genome sequencing, methylation, RNA-seq, global proteomics, phosphoproteomics, and glycoproteomics. We've targeted proteomics. We also have targeted proteomics, SRM and MRM, and single-cell RNA data samples for a subset. A major aim of the study is to study adolescent and young adult in the larger context of human development. We've therefore merged our proteogenomic data with that from 99 adult GBM tumors from another CPTAC discovery cohort. For our survival analysis, we've collected mutation and clinical and survival data for over 1,700 tumors from multiple previous studies. We're also making use of external data on protein and gene expression from normal tissue over the lifespan. Finally, we have a validation data set including 29 pediatric and adolescent high-grade gliomas. The HOPE cohort itself includes clinical data from 93 events across 86 patients. This breaks down into 2 groups: 59 pediatric patients, ages 0 to 15, and 34 adolescent young adults, ages 15 to 40. Leveraging external data from this large cohort, we've characterized age-dependent mutation patterns. On the left, we see 2 mutation groups. Group 1 mutations like TP53 and ATRX peak in the young adult, while group 2 mutations like PERT and P10 increase in older patients. We see lower frequencies of mutations in the pediatric groups overall compared to other age groups. The bubble plot on the right highlights how the prognostic impact of mutation varies by age, with some mutations like TP53 and H3F3A being more impactful in pediatric patients, while others, including P10 and IDH, are more impactful in older patients. The genomic landscape differs by age group. On the left, we identified a group of genes like CRYBG1 and IGFB23 that are differentially methylated depending on age group. Protein and RNA expression of these same genes are anti-correlated with methylation, suggesting age-dependent epigenetic regulation of these molecules. On the right, copy number variations also differ between patients under and over 15 years of age, revealing distinct genomic alterations. To study age effects using proteogenomic data, I mentioned that we merged 93 samples from the HOPE study cohort, which spans ages 0 to 40, with data from a previous CPTAC GBM study focused on older populations. This allowed us to model protein and other molecular expression across the entire developmental lifespan. We've analyzed the temporal profiles of various omic types. These temporal profiles are just localized regression fits based on the age at diagnosis, as shown on the left for CN-TN1, which shows an increasing trend in abundance over time. To identify age-related patterns in the molecular landscape, we performed multi-omic clustering of these profiles, illustrated in the middle of the heat map. This revealed a notable shift around age 26, right in the middle of the adolescent and young adult age group. This led us to redefine this group into two distinct subgroups, adolescent and young adult, ages 15 to 26. Using our large external reference cohort of mutation and survival data, we were further able to establish that these two groups showed significant differences in survival outcome, with the young adult group having the very best survival. Additionally, kinase and transcription activities vary between these new age strata. For example, the first box in the upper left highlights a set of kinases related to microtubule regulation. These kinases activate different sets of transcription factors. Pediatric patients here are highlighted in blue and adolescent in red. I mentioned that we leveraged external data from normal tissues. Specifically, we've used this data to model temporal expression profiles of normal proteins and transcripts collected from normal tissue samples. By systematically comparing age-dependent profiles between normal and tumor tissues, we analyzed their differences using pathway enrichment analysis. Pathways where tumor expression is higher are shown in orange, and while those with lower expressions are shown in purple. One key finding is the reversal of oxidative phosphorylation activity. In pediatric cases, OXPHOS is lower than normal, but in the young adult, it's higher than normal. This pattern is consistent for both males and females. A similar reversal is seen in the neuronal system pathways, though this pattern is only significant in males. I mentioned that we're interested in understanding how age-dependent tumor molecular profiles impact survival and in identifying age group-specific effects, especially those affecting the adolescent and young adult group. However, even after merging our HOPE data and our GBM data, our cohort of 156 patients remains for this kind of complicated analysis. To address this, we introduced a novel transpopulation analysis approach that leverages our external reference data on mutation, survival, and age. By interpolating the estimated tumor molecular profile using the age of each reference sample, we can predict the abundance of each protein for those reference samples. This allows us to perform survival analysis on a cohort of thousands of glioma patients to identify proteins and pathways associated with survival. Importantly, since the survival and mutation data from our study cohort is not used in identifying survival-associated markers, our study data can be used to verify these findings. In essence, survival inference within the larger cohort acts as a data reduction method, enabling validation within our smaller cohort. This heat map summarizes the transpopulation survival analysis results, highlighting key pathways and constituent proteins associated with survival outcomes. The squares are indicating concordant associations where both the study and the reference population show consistent results. Among other pathways, oxidative phosphorylation is robustly associated with better survival, where the pattern is particularly strong in males. We're especially focused on identifying biomarkers particularly relevant to the adolescent and young adult period, and we've identified two key markers with robust and reproducible associations, including GRID2 and PHKG2. Here, we're further showing validation of these two markers using data from the independent cohort of 29 samples. PHKG2 is associated with better survival. It's involved in glycogen metabolism, and it specifically regulates glycogen phosphorylase, a key enzyme in glucose mobilization. GRID2 is a glutamate receptor associated with poorer survival and involved in synaptic transmission. Using this transpopulation survival analysis framework, we identified 150 kinases associated with overall survival in the adolescent and young adult. This is actually the first step in a broader analysis aimed at finding potential drug targets. Next, we performed causal network analysis to identify kinases with a direct influence on prognosis. Interactions among these proteins were validated by comparing cancer versus normal interactions and by evaluating interaction scores on a per sample basis through gene perturbation experiments. These steps help to prioritize candidates for follow-up cell line experiments. We hypothesized that targeting kinases associated with poor survival may benefit the subset of patients in whom these kinases are active. By inhibiting kinases whose activity was associated with poor prognosis in a drug or CRISPR knockout experiment, we expected to see greater suppression of cell proliferation in the cell lines with higher kinase activity. Indeed, using preliminary CRISPR and drug response data from our cell line perturbation experiments, we observed strong concordance with this hypothesis for the 6 putative causal kinases identified in our analysis. We therefore suggest that these kinases are potential therapeutic targets and warrant further investigation in follow-up experiments. We've also made a systematic effort to investigate cis and trans regulation in the tumor. In a trans-regulatory analysis, we observed significant impacts of IDH1 mutation, particularly in the methylation landscape, which is well documented. IDH1 mutations are known to alter DNA methylation patterns by producing proteins that inhibit DNA methylation. We observed that this hypermethylation alters gene expression and protein expression, affecting pathways related to immune function and glycosylation, among others. By further analyzing our methylation data in conjunction with protein and RNA within a mediation framework, we were able to explore the mechanisms by which IDH1 mutations drive these downstream effects. In this way, we identified several proteins that mediate the effects of IDH1 mutations on methylation, as shown here. We also performed sex-stratified consensus clustering of protein pathway scores and identified 3 distinct clusters of high-grade gliomas in each sex. On the left, you can see the single-cell GRCA matrix of the top associated pathways, and on the right, the kinases enriched in each subtype. We have 3 clusters. The first cluster is enriched of pathways involved in RNA processing and metabolism. The second cluster with immune and inflammation-related pathways, such as complement and cytokine signaling. And the third is enriched with neuronal and energy metabolism, where we see high expression of OXPHOS. These clustering results were replicated in an independent cohort of 29 samples. Several of our key causal kinases are enriched in these subtypes, including ATM in cluster 1, LCK in cluster 2, and NTRK2 in cluster 3. ATM and LCK were identified as drug targets through the drug target pipeline I just mentioned. And their enrichment within these clusters helps to identify appropriate target populations for therapies targeting these drugs. The motivation for performing sex-specific consensus clustering came from an interesting observation. Samples in cluster 2 enriched in immune-related pathways show strikingly different survival outcomes depending on sex. To better understand what might contribute to such differences in survival for the subtype, we performed pathway analysis for global protein data. We found female-biased expression of interferon gamma, an alpha response, and male-biased expression of collagen formation and EMT pathways. To identify molecules that might explain the sex-specific survival outcomes seen in cluster 2, we looked for proteins specifically upregulated in males as compared to females specific to this cluster. We identified PLA2G4A, a phospholipase known for its role in platelet activation and a member of the otherwise female-biased interferon gamma pathway, as being specific to males. Additionally, we observed an association between this LCK, one of our causal kinases, and PLA2G4A expression, motivated by our observation of sex differences in biological processes in which glycosylation play a key role. We characterize cluster 2 specific sex differences specifically using our glycosylation data. We found several pathways, including integrin signaling, were uniquely enriched in males in this domain, but were not detectable based on RNA or protein. Specifically, we identified a glycomodification enriched in males in cluster 2 that featured both fucosylated and sialylated modification. Interestingly, this glycomodification is negatively associated with PD-L1 expression, but only in males, suggesting a sex-specific role in immunomodulation. We made use of single-cell data to identify cell type-specific markers for basic immune cell types and tumor-associated cells within the myeloid compartment. This served as the basis for multi-omic deconvolution analysis, which was performed to estimate the fraction of each cell type in the tumor. Overall, we observed a lot of heterogeneity in cell fractions across samples within this study. We found that different types of immune cells, including CEP1, monocyte tumor-associated macrophage, and T cells were more enriched in this M2F2, enriched of inflammatory pathways. In addition, we noticed a correlation between age and the infiltration of different cell types. For instance, CEP and T cells were less enriched in pediatric tumors compared to adults. We observed that female patients with high CEP and low T cell fractions were associated with worse prognosis, even after adjusting for age and H3F3A mutation in patients under 40 years of age. This observation is consistent with previous literature reporting that CEP monocyte-derived TAM cell type is more enriched in recurrent and more aggressive tumors. This finding was replicated in our validation cohort as well. We observed intriguing variations across age and sex groups in the associations between T cell and CEP percentages with immune markers and also with our M2 specific markers. For example, PD-L1 showed a positive correlation with T cells only in adult tumors, but not in tumors from patients under 40. PD-1 showed a positive correlation with T cells in both sexes in adult tumors, but only in females under 40. In parallel, we observed a significant association between T cell and CEP cell percentages and M2 markers, including ITGAV and PLA2G4A. By leveraging the single-cell data, we gained a bit more information about the cell types in which these genes are expressed. ITGAV is expressed in both tumor and non-tumor cells, while PLA2G4A is expressed at the highest levels in the blood vessels and myeloid cells. Altogether, these observations suggest that distinct immune suppression mechanisms are at play in HGG tumors across different age and sex groups, and therefore tailoring treatments to each age and sex could lead to improved efficacy and advance the goals of precision medicine. With that, I would like to again acknowledge the efforts of my many colleagues and collaborators at Mount Sinai, at CPTAC and CPTN and beyond. Thank you, and I'm happy to take any comments or questions.
While you all are thinking, I'll just reflect that during this analytical journey, and a journey it has been, one of the sort of the most surprising things for me anyway was you always try and correlate everything to survival as a way to have like a real world touchstone for some of these findings, and how that was just so incredibly difficult, and we weren't really seeing signal there until we separated men and women or males and females apart, and then it all just kind of clicked. So just underscoring what you showed, that there's so much real important biology that is sex-based. Javed.
Right, and I think we discussed this with Adam yesterday. There is a wealth of proteomics data that has been now generated through CPTAC, through your efforts. Mandy has done this for liquid biome. We have associated genomic and RNA data for some of these cases. There's also effort here for spatial proteome mapping, as we saw earlier for AI, which seems to be much more advanced than the actual proteomics, because that could be clinically relevant. If you take a tissue from a patient and can do an AI to read or an immunohisto or multiplexing to see the concentration or the presence of proteins in their tissue, versus the actual proteomics that one requires a lot of tissue, second takes a lot of time, third is prone to be degraded. Phosphoproteomics, for example, is not always doable. Are there any possibility to merge the existing proteomics data that we have to the genomic in a way that we could predict the presence of the proteins in a sense that we don't really actually have to do the proteomics, which is again a lengthy. I think Adam is getting my point to this, but because I think to make this clinically relevant, we also have to make it fast. We have the genomic data and RNA that we could produce fast, but proteomics is not there, and it will require so much tissue that we often don't have.
So, do you have thoughts on that, Nicole, or you want me to? Let me take a stab at it and see if you want to add to it. So I think the thing about proteomics really is one of the key things that makes it possible to derive biological signal from it is the depth, right? And so what you can get from tissue and mass spec is the depth and the post-translational modifications and isoforms and all of that, which you can't really get with some of the platforms you're measuring. But you're right that we need to be able to then link this back to a more, clinically actionable realm. And I think that's where signatures come in. And so, Nicole, you mentioned targeted proteomics, and that's like, as Henry showed, that we're integrating the proteomic signature into the craniopharyngioma PNOC trial. That's really just based on immuno-MRM on a signature panel for a particular pathway that's being targeted in that trial. And so I think when we start finding these risk groups, and we're able to extract a signature in a data layer that is more accessible, and that becomes a more clinically useful tool. So you have to kind of go deep before you can resurface and I think create something. That'll actually be a theme in the next talk as well. Did you want to add anything to that? I think we're good.
I was just going to say one of the things that we saw, for example, with the Raman spectroscopy talk, right? That, and I don't know if people are doing this in CPTAC yet, right? But this histology-based mapping to proteomic features or imaging-based, mapping to proteomic features. So doing the same type of AI machine learning frameworks that, go all the way through. I think that's sort of what Javad is thinking about, but starting with clinically available modalities as a context.
Thank you, Nicole.